Hey everybody, in this video I'm going to discuss the Krebs cycle. The Krebs cycle is the step that occurs after the intermediate step where we have two acetyl-CoAs that will go through a series of oxidations to eventually produce NADHs and FADHs for the electron transport chain. As we can see here, this is a picture of the Krebs cycle, and as you are looking at this diagram, you will start at the top with the orange rectangle that says acetyl-CoA. This is where the acetyl-CoA in the intermediate step will enter. As we can see, there are lots of chemical reactions that are taking place in the Krebs cycle. Each one of these chemical reactions is considered a redox reaction, which, if we remember from the first week, means that as one thing is becoming oxidized, something else is becoming reduced. As we go through all of these steps, we will see that we are making NADHs and FADHs that will eventually go to the electron transport chain. If we look at the example of isocitrate to alpha-ketoglutarate, we will see that as isocitrate is being oxidized to alpha-ketoglutarate, NADH plus is being reduced to NADH. The same type of thing happens later on in the cycle when we go from succinate to fumarate. As succinate oxidizes to fumarate, FAD is going to be reduced to FADH2. This is a great example to show how redox reactions occur. In terms of what you need to know for your exam, I do not expect you to know each individual step of the Krebs cycle, but I do want you to recognize that it's a series of redox reactions and know what we're getting out of the cycle. In terms of enzyme regulation, isocitrate dehydrogenase is the rate-limiting enzyme. As we remember from our discussion on enzymes, a rate-limiting enzyme is the enzyme that's going to control the whole process. So even though isocitrate dehydrogenase is the enzyme that makes alpha-ketoglutarate from isocitrate, this enzyme is also going to control the whole process. All of the enzymes of the Krebs cycle are regulated in the same way. For example, they will all be product inhibited. What this means is that if there is already enough product in that particular chemical reaction, the enzyme is going to slow down. So if we go back to our diagram here and we take the example of citrate to isocitrate, an enzyme is product inhibited, meaning if there is a lot of isocitrate, that chemical reaction isn't going to occur as quickly because we don't need more of it. Enzymes are also going to depend on substrate availability. This means that there has to be enough of the substrate in the chemical reaction for it to occur. So if we look at another example on the other side of the cycle, let's look at fumarate to malate. There needs to be enough fumarate in order for the chemical reaction to occur for it to go to malate. If there's not a lot of fumarate available, then that chemical reaction cannot occur. Lastly, all of the enzymes are going to be inhibited by high NADH, high ATP, and low acetyl-CoA. This makes sense because the goal of the Krebs cycle is to make NADHs and FADHs. If we already have a lot of NADHs that are going to the electron transport chain, we don't necessarily have to make more. So therefore, the Krebs cycle can slow down. Also, it makes sense that the enzymes would be inhibited by high ATP. If we already have a lot of energy available, we don't necessarily need to make more stuff that can make ATP. And finally, enzymes will be inhibited by low acetyl-CoA. And we can relate this directly to substrate availability. Acetyl-CoA is what's entering the Krebs cycle. If we don't have a lot of acetyl-CoA that's coming in, obviously the Krebs cycle can't occur as quickly. Finally, the last important thing to know is what we get out of the Krebs cycle per turn and per glucose molecule. As you can see from the diagram, for every turn of the cycle, we're going to get 3 NADHs, 1 FADH2, and 1 GTP. The 3 NADHs and FADH2s go straight to the electron transport chain. The GTP goes through a quick chemical reaction to make ATP, and that can be either stored or broken down directly for energy. This is describing what happens for one turn. But for a glucose molecule, we know that we make 2 pyruvates at the end of glycolysis, which then go to 2 acetyl-CoAs, which enter the Krebs cycle. Therefore, we will actually go through the Krebs cycle twice. So per glucose molecule, we will actually get a gain of 6 NADHs, because 3 times 2 is 6, 2 FADH2s, and 2 ATPs. This is the end of what you need to know about the Krebs cycle. The next step that we will talk about is the electron transport chain, which is where the NADHs and FADH2s are going, and that will be discussed in a separate video.